which he does often do, and sitting in his bedroom playing with a molecule of DNA, it seems. And in walks his mom, and she says, "What you doing, honey?" in her Southern Texas drawl. And he says, "Hypothesizing about the shape of nucleic acids in a sulfur-based life form. I'm sorry, a silicon-based life form." And then she makes some comment about evolution, and then they move on with whatever the sulking story is. I can't recall. That show is remarkably scientifically accurate, which is one of the reasons why I like it so much. They actually keep physicists on staff to make sure they don't say things that are incorrect. And so the selection of silicone as the alternative life form is no accident. Okay? Silicone, if we were to have another life form, is our closest cousin to carbon. Because remember we talked last class, or 2 classes ago, about periodic table and how all of the elements that are in the same column have the same number of valence electrons and, by logical consequence, the same chemical properties. Well, what is the closest element to carbon? Well, that's going to be silicone. So what makes carbon so special? Well, a couple of different things. First and foremost, it's everywhere. Carbon is really, really common here on the planet. And so it is really abundant, easily available. The second thing that makes carbon so important, and that makes silicone the logical, although we've never found a silicone-based life form, so let's not jump to conclusions, but the logical second choice is that carbon has how many valence electrons? 4, right? If you didn't notice the Lewis dot, 1, 2, 3, 4. That gives carbon a huge amount of versatility when it comes to building molecules, because it can be bonded to itself, it can be bonded to other things, it can engage in single bonds and double bonds and triple bonds, it can form rings, it can form chains, it can form all sorts of stuff. And so carbon having 4 valence electrons makes it an ideal candidate to build large biological molecules. And that's going to be the topic of today's lecture. This is everything I just said up top. Today we're going to talk about the 4 different kinds of large biological molecules. When we think about what those 4 kinds are, the first 3 are really easy. It's what gives you calories when you eat food. Okay? Carbs, fats, and proteins. Now, instead of calling them fats, we actually call them lipids, because there's some other stuff that's in that category. Okay? And then the 4th large biological molecule is the one that's in the nucleus of all of your cells, and that is DNA, nucleic acids, right? So nucleic acids would include both DNA and RNA, right? So when we talk about these large biological molecules, today we're going to talk about some generalizations, things that are true about all of them, and then we'll take turns and look at each one at a time to see how each is unique and the roles that it plays in your body. So we'll talk about carbs and lipids and nucleic acids today, and then the packet I'm going to give you at the end of class is to kind of very gently introduce you to proteins, and the next class will be all proteins. There's a ton of practice work in your workbook about these large biological molecules, mostly because I really am bad at chemistry, and so it took me like 40 pages of practice before I figured it out, and so I gave you like 40 pages of practice in order to figure it out. I am not going to collect any of it. I'll post answer keys online, okay? The goal, I mean, you can go online and copy it if you want, but that's really not going to help you learn. The goal, of course, is for you to try it, figure it out. You really just have to spend time working with the content in order to learn it. Every once in a while, I come across a student who's just brilliant and has it figured out, but we don't know if that's you until you take the test, right? So don't bank on it. So that's kind of the direction we're going, and my encouragement to please do the practice, and I'll show you where that is in your workbook a little bit later on. Okay, so what makes carbon so special? This is what I was saying earlier. It's readily abundant. Readily abundant, and has those 4 valence electrons. Those 4 valence electrons are super important because that means we can form these single bonds, these double bonds, all those different bonds. And due to its small size, so carbon is a small molecule, the bonds that it forms are actually very stable. Okay, yes?
You know how they have like the valence ring?
Uh-huh.
If you have more valence rings, is it like, does it affect
Well,
the
valence
bonding?
is just the outside, right? Valence is the valence shell. So you have energy levels.
Yeah, the energy levels.
So the further away they are, remember, this is So that's
More
more
potential.
potential energy, right? So
Yeah.
that's where it comes into play. But because we're so far away from the nucleus, and the nucleus is the positive and the electrons are the negative, those bonds aren't as strong,
Okay.
right? So the small size of carbon's really little, right?
Yeah.
Because it's got that one energy level. Carbon is really little, and so because of that, the bonds that do form are really, really stable.
Okay.
Now, as you recall from chemistry at Ho, whoops, wrong direction, you can form a bond on each side, right? More or less. So we can actually make 4 bonds, or we can make 2 bonds here, double bond here, and double bond there. Or we can do a double bond on one side and then single bonds on the 2 other sides. So because there's these 4 valence electrons, there's a ton of versatility to that carbon atom. And very directly related to that, it allows a lot of different branching to happen, and that makes carbon really versatile. As a result, we can make these chains, right? We can take, oops, I got a new clicker and the buttons are in a different order. This is messing with me. What we can do is say carbon is going to join another carbon, 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 and that can go on forever, right? There's no upper limit to the chains that we can build out of these carbon molecules. And so when it comes to carbon, they are the skeletons of almost all of our organic molecules. And so that's why we are a carbon-based life form, because carbon is the basis of all of these different molecules. And so these carbon chains can vary based on length, whether and where double bonds are, whether and where we branch, and even we can form circles, rings. This is true for carbon because of those 4 valence electrons. Okay? A lot of variety. So all of these things on the previous slide, see how they're made of just carbon and hydrogen? Those are called hydrocarbons. We love that name, hydrocarbons. So we can take any one of these hydrogens, any of them, and replace it with a chemical group. Okay? Hydroxyl, carbonyl. You replace it with a chemical group. And when we do that, we start to produce unique molecules. Molecules that have very unique behaviors, very unique shape. And I'm going to connect that back to the quad we did a couple nights ago. Remember, shape is like the fingerprint of a molecule. When we start adding these functional groups, now we're starting to make these large biological molecules that have very unique shapes. And if they have unique shapes, they have unique functions. Okay? So chemical groups, that's what this is saying here. Distinctive properties of those organic molecules depend on the arrangement of the carbon skeleton, as well as the number and arrangement of the groups attached to that skeleton. So what we've been looking at were just the skeletons. Now we can add stuff to the skeletons in order to produce these unique molecules. Again, the chemical group can replace one or many of the hydrogen atoms. So functional groups play a role in chemical reactions. Not all chemical groups are functional groups, even though those 2 words are often used interchangeably. One of the chemical groups you will learn, of the ones I'm going to ask you to learn, only methyl is not functional. It turns out that there are hundreds of different chemical groups. And if you took into chem, you got to learn many of them. I'm only going to ask you guys to learn 7. 6 of them are functional, meaning that they are engaging in chemical reactions. The last, methyl, is not reactive, so we call it a chemical group. It's used to tag things. It's used to modify DNA and the activity of different genes. We'll deal with those later. You do need to memorize the 7, okay? But I'm going to go ahead and try to make this not real tricky. Do not, under any circumstances, memorize examples. That's not necessary. But when we look at this, okay. Oops. Hydroxyl, hydrogen, and oxygen. Hydroxyl. All right, we can do this. Oh, yeah. All right. Carbonyl, carbon, and oxygen. Okay. This is where kids get confused, carbonyl and carboxyl. But carboxyl is both of them together. Carboxyl is carbonyl, see it, plus the hydroxyl, see it? Oh, so carboxyl, it's got both of them, right? So it's got the hydroxyl and the carbonyl together. Then we have an amino group. You know, an amino group has nitrogen in it. We find that in an amino acid. See, we like it when the names make sense. A sulfhydryl group is the one that has sulfur. The phosphate group is the one that has phosphorus. You guys got this? Right? So I am asking you to memorize 7 of them. Methyl is the one that is not a functional group, just a chemical group, and it's your CH3. So I don't have a trick for that, but there you go. Yeah?
How memorized do you have to have them? Like,
You're going to have to use them on your test. Like,
So
I'm never going to ask
so if
you.
you just say like phosphate group, I gotta know.
Yep.
And like that
So it's not just like matching like phosphate group.
No,
the one
that
with
is
the P.
CP Bio at its finest. I'm never going to ask you to please draw a phosphate group. Not going to happen. You're going to have to be able to use the properties of that group in context.
Okay.
What are the properties of a phosphate group? Polar or non-polar? What is it?
Oh,
Polar, what?
it's negative.
And it's functional. What else? What identity? Oxygen. Oxygen is extremely what? Yep, highly electronegative. So the presence of oxygens in a phosphate group is what's going to make that polar. Hydrophilic, hydrophobic? Which one? If it's polar, it's hydrophilic. Those are the kinds of things you're going to need to be able to know. Oh man, right?
Yeah.
So we have graduated from honors bio where it's like, here, draw hydroxyl. Cool, A+. No, you got to be able to use in context.
Yeah.
Okay, that's going to be the big step between AP and honors. But you got this. You'll get used to it. Do what?
Can you go back
For what?
to hydroxyl to amino, just the names?
So this is straight out of your textbook, okay?
Is that on Schoology?
No, everything's on Schoology. Okay, so now macromolecules. Macromolecules are polymers. Poly means what? Many, and we built those things from monomers. Monomers mean one. We also have these things called dimers. Di means two. Okay? So in a way, my caterpillar kind of does relate. In a way, my caterpillar, you can think of it like a polymer, right? Where each one of these is a monomer. 1, 2, 3, right? So they're all linked together. And in biology, the only kind of bond that's really useful is the covalent bond. So we have to link together our monomers by covalent bonds. But each different kind of polymer has a special kind of covalent bonds. We just give it a fancy name. Okay? So each kind of polymer has a fancy name, covalent bond that holds its monomers together. So how these things are put together is essentially the same. We like that. We like it when one kind of process matches a bunch of different things. Monomers are connected to each other by what we call a dehydration synthesis. Okay, take chemistry. What is synthesis? A synthesis reaction is when we combine multiple things into one. Okay? Dehydration means we are removing water. So here we are taking two smaller things, synthesizing them into one larger thing, and in doing so we remove a water molecule. That's dehydration synthesis. When we do dehydration synthesis, we produce a covalent bond. Okay, a covalent bond. The name of that bond will be specified as we go through and look at our different macromolecules. Okay, so this is dehydration synthesis. And when we do this backward, let's say we want to break our caterpillar friend apart, we disassemble them in reverse order. We have to break a covalent bond by inserting a water molecule. Okay, inserting a water molecule. When we do this, we are going through what we call hydrolysis. What does lysis mean? Something lysis. What does that mean? A cell lysis comes apart. Right? To lyse comes apart. Cells. That's what it means. So when we go through hydrolysis, we're cutting this apart by using water. We added a water molecule in order to cut it apart. We broke that covalent bond. Digestion is a great example of hydrolysis at work. You eat macromolecules. Remember also what we said, they're on the label, on the side of the food that you eat. You eat macromolecules. That's great, but in order for your body to make use of these things you're eating, they have to be either monomers or dimers. Okay, they have to be much, much, much smaller. So the job of digestion is actually to take the macromolecules, the polymers, and break them into monomers. So when people tell you you need to drink water, it will help you digest your food, They are right. Because look at what's happening. Water has to actually be inserted to break every single one of those covalent bonds. Water does help digestion. Okay? Life skill. Look at us. On a roll. Polymers are incredibly diverse. Obviously, because polymers could be fats, proteins, nucleic acids, carbohydrates. Polymers are incredibly diverse. Similar to using letters of the alphabet, with 26 different letters in the alphabet, we can form literally hundreds of thousands of words of various lengths, right? You can have short words like as, or slightly longer, ask, slightly longer, astute, right? Have really long supercalifragilisticexpialidocious, right? So how you assemble those letters can have a short word or a long word or a really long word, and all of those words have completely different meanings based on the order of the letters. The same thing is true when we look at polymers. Okay? Polymers have different roles, different functions in organisms based on how those monomers are put together. And that's never more important than when we get to proteins next class. Okay. We're going to start by looking at carbohydrates. I actually think carbohydrates are the hardest. Sorry. But that's the one we hit first. So carbohydrates, first of all, if you think, this is all brand new. I took honors bio and nobody taught me anything. I feel so sad. I want to show you, I hear that all the time. They didn't teach me this in honors bio. I literally went to an honors bio teacher and got, this is what your notes look like on macromolecules when you took honors bio. You have the grid, you fill them in. Yeah, this is Ms. Hampton's handwriting. Yep. So, you should have had something that you may not have had this particular grid, but it was on your common assessment, so which means no matter who you had, you got to learn about macromolecules. And I'm like, I blocked it from my soul. I wish no recollection from honors biology at all. I hear that a lot. It's okay. So,
2 years ago.
you're like, yes, you know how much I've done since honors bio. So if you took honors bio, you should have covered macromolecules. I can't say definitively that it happened because I wasn't there, but it was on your common assessment. So presumably you covered it at least a little bit. So hopefully this comes back to you pretty quickly. And if it doesn't, plenty of reading, plenty of practice to get you up to speed. Carbohydrates, the names end in OSE, O-S-E.
Like glucose?
Okay. Like glucose, correct. Or sucrose. Or lactose.
Oh. Isn't that neat? They just
I would
keep
start
asking
making a
for
list of all the things we learned here. Usually we get to the first quarter and kids are like, I think I've learned more in 9 weeks with you than like my entire life. I'm like, oh, that's fine. Catch me up. All right, so monosaccharides, mono means one, is the monomer, right? So we put together bunches of monosaccharides in order to make polymers or polysaccharides. Okay, so saccharide is just sugar. Most of the monosaccharides have this base unit, CH2O, any multiple of this. So we could have C6H12O6. Okay, or a pentose is C5H10O5.
Oh, so just kind of follow that.
Yeah, exactly. So
ratio.
if you see this ratio, that is a monomer of a carbohydrate. Okay? Look for the ratio.
Ooh,
Okay?
so it's still a monomer? Even though
Well, this is a monomer. These are all monomers.
it's
Okay? Right? Like, oh, man. It's all right. Notice how when we have in its linear form, we count from our carbon up top, 1 through 6. We always count our carbons. We like to count our carbons because then they're easier to identify. Oh, I added a functional group off of the 4th carbon. What's that? Okay. So here's what happens kind of wonky with carbs, is that we move or we flow between linear form, and then we can just for fun rearrange into a ring form. Okay? Just for fun. This doesn't take an addition of energy, it just changes. Okay? That's fine. It's really important because in order to undergo dehydration synthesis, in order to have a monomer form a dimer, we have to be in our ring form. They really very rarely exist in this form unless you're digesting them. Okay, that's when we get into cellular respiration, that's when we tend to have them in their linear forms. This is far, far, far more typical. The only reason I want you to know that they stretch out is because when I start showing you this later, I want you to recognize that it actually is glucose. Yes?
So, it's still a monomer even though there's multiple
All monomers.
CH2Os?
Yep.
So like, would it have to, like,
That's a dimer.
so it connects with
Another one,
another one of the,
and that's a polymer.
oh, okay, well, I'm not there yet.
I know, that's why, but I thought I would just show you. Ah! Ah! Go back to one!
So, what's it connect to something that's not the same as a C6H?
So multiple monomers can connect together, they don't have to all be the same. Right?
But
So
it's still a monomer even though
it's
there's,
still a monomer. Because it's like there's 26 different letters in the alphabet, right? Each letter is a different monomer. Okay? Anything that has this ratio, CH2O, is a monomer of a carbohydrate.
okay,
Okay? And you're going to join the monomers. They can be the same or they can be different. When
so,
we put them together, that's how we make a macromolecule.
so like, say you put like the C6H12O6 with like a C5H10O5, then you get a,
Then you get table sugar. That's this one.
okay. Dang, I just made sugar.
Well, the guy did it, but you can type it if you want. Do you see what I mean?
Yeah. And so the polymer is multiple of them put together.
So you've heard of these. What's the difference between a simple and a complex carbohydrate? Oh, simple, complex.
Oh, okay, so, yeah.
Rock your world.
All right, thank you for helping that, 'cause I was confused.
Okay. You guys have heard of the right to avoid the simple sugars, right? Simple sugars spike real quick.
Oh, yeah.
Spike your sugar real quick. Well, what are your simples? Yeah, it's this stuff, right? Do you have to do a lot of work to digest it? No, because it's pretty much already broken down. Let's say instead you're going to eat something like that, you got to work to digest that. Oh yeah, that's a complex carbohydrate.
So why is that better for you? Taking more time to digest?
Because it doesn't cause the huge spike in blood sugar followed by the huge fall. It's a more slow release of energy into your bloodstream.
Oh, interesting.
Yeah, look at this, life skills and life
Put that on the list. Learn nothing.
You are like, look at this, life skills, yes!
So like, does it break down a lot slower? Because like, people say like before like a competition day you're supposed to eat like complex carbohydrates. So like, does it take that whole time to break down?
I don't think so.
Oh.
I don't think so. I still think you're digesting in a matter of hours, but it's as opposed to a matter of minutes.
Okay.
Right? Because that simple stuff, it hits your stomach and it hits your blood like 20 minutes, real quick. The complex stuff takes much longer. But no, it's not days. It's not days. I want to say the new research, and you guys should correct me because I'm not an athlete, in case you were in doubt. I think that now they're saying if you're trying to eat the day before, I think you're supposed to eat protein as opposed to carbs, complex carbs. Yeah. So I don't know. Listen to your personal trainer if such a person exists. Okay, so when we put together monomers, guys, it's the same reaction. Remember I told you dehydration reaction is how we make these polymers. And I told you that when we make those covalent bonds, we give them fancy names. Okay, a fancy name for this one is a glycosidic linkage. Glycosidic. It's just a
So-
fancy covalent bond.
So a dimer is only two.
Dimer is two. What's that?
Do we need to know the name?
Yep. Yes, you do. You get to know the names. I'm going to give you a fun little organizational chart for you to fill out.
Good.
Okay? Glycosidic linkage. We can make a whole bunch of those glycosidic linkages,
And that'll give you
and that gives you a
Potato.
complex carbohydrate. That'll give you a potato. So when we build these polysaccharides and polymers, polysaccharides can actually do one of two things. They can either do structure or storage, in essence. So energy storage, that's typically what we think of with carbs. So starch in plants. Plants store their carbs as starch. Have you ever eaten a green banana? Right? Yeah, it's that'll leave a film in your mouth, right? The reason it tastes like that is because it's starchy. Plants store their carbs as starch. As they ripen, well, they ripen after they've left the tree. As they ripen, they break down. They break down into fats. So a ripe fruit tastes sweet.
So how come? Like,
So green bananas, unripe fruit, is actually way healthier for you than ripe. It's just so, like, it's disgusting. That's correct.
it's a grow you banana treat now.
Right? Or you could just go shopping, either way. So do you see how like all of these kind of come together? You're related to food. You've experienced these things before. You just didn't realize what was happening in terms of macromolecules. So energy storage, starch in plants. I don't store starch, okay, but I need to store carbohydrates. I store it in the form of glycogen. I can store glycogen in my liver. I can store it in my muscles. If I'm full up on glycogen, then my body says, oh, bonus glycogen, we're going to turn that into fat, right? And so we have to morph it into the lipid version, right? We'll talk about that and talk about insulin and glucagon and all that kind of stuff later on in the year. But I can store stuff too, I just store it in a different way, okay? Let's see, that's fine. The other thing that can happen with carbohydrates is they can be structural, okay? Structural polysaccharides. Cellulose in plants, so that's like wood on a plant, right? Leaves. It's carb-based, okay? But it's cellulose. Cellulose we know as fiber. You can't digest it. Okay? It passes right through you. Fiber's really good for you. Okay? It will matter more to you in about 30 years. But invertebrates and many fungi also have structural polysaccharides. You don't have any in your body. But when you step on a gigantic crusty spider, oh, so you make that noise.
I hate that.
Right, you know that noise?
I can't do it. Oh, I feel
Oh,
trapped.
Rogers, why? We were doing so well this morning. Oh, well, and so you had to destroy its chitinous exoskeleton. Chitin is a structural polysaccharide. Okay? So the exoskeletons of invertebrates, okay, is also polysaccharide structural. So how do they do that? How do you have a polysaccharide that could either store energy or be an exoskeleton? Well, it has to do with our bonds. And I'm sorry, but we have to look at these one more time. So this was our glucose, right? C6H12O6. That's what we said earlier is our glucose. All we have to do is flip where the OH is, okay, to switch between what we call an alpha glucose and a beta glucose. Okay. Just that one switch, when we start to put them together into polysaccharides, we'll say, oh, here when all of our OHs are on the same side, that's starch. And when we have them on opposite sides, that's cellulose. So how those monomers connect is going to be the difference between the storage and the structure. Okay, so storage and structure. Okay.
Is that like, no one knows what other bonds change, it's just the location. And
Well, it's the whole thing is flipped over. Right, so do you see how we have the CH2OH on top? And then here they're switching. The whole thing is just flipped over,
Interesting.
right? And that one difference is enough to switch between storage and structure. Let's talk lipids. So lipids, we call them fats earlier. Lipids is an umbrella term that includes things like fats, waxes, phospholipids, steroids, and hormones. Actually fit into this category as well. Lipids are not true polymers because there's an upper limit to size. So I told you earlier that what makes a polymer is that we can keep adding monomers over and over, and monomer, just keep adding them onto the end, right? We just keep having the dehydration synthesis, and we just get a bigger and bigger molecule every single time. Well, lipids are not true molecules because they have an upper limit to size. They just can't keep growing forever. And that upper limit makes it a large biological molecule, but not a true polymer, okay? So that's going to be the distinction. Some people say they're not large enough to be considered true macromolecules. Different textbooks use different language. I'm not really worried about it. I need to make sure that you understand what a lipid is. So compounds that are called lipids are grouped together for one property only, hydrophobic behavior. So what we're going to see in lipids is a lot of hydrocarbons. Hydrocarbons are non-polar bonds. Okay? So anytime you see a molecule that has a congregation of those carbon and hydrogen bonds, okay, that's going to be nonpolar, that's going to be hydrophobic. And what we're going to get to in just a second here is we're going to start to see some of these molecules that are so big that a region of the molecule can be very heavily hydrocarbon and nonpolar, and a region of the molecule can actually have an oxygen mixed in, and that region could be polar and hydrophilic. So regions of the molecules can we call it amphipathic. There we go, amphipathic, which means the two different regions of the molecules have different polarities and different affinities to water. So when we look at lipids, okay, lipids start with glycerol. This thing is called the glycerol. And the presence of the glycerol is what gives this an upper boundary. Because at each one of these carbons on the glycerol is a location for dehydration synthesis to occur. And when this occurs, we're pulling out the water and we're attaching a hydrocarbon chain to the end. We call this thing a fatty acid. Okay? A fatty acid. Note all this nonpolar, okay, hydrophobic. We're going to lose some of this. We're going to have a little bit of polarity here. For the most part, the molecule itself is overwhelmed by those nonpolar hydrophobic bonds. Okay. So, we have our glycerol, and we have our fatty acid chain. They can join together at that carbon here. We call that an ester linkage. So that's the fancy name of this covalent bond, is an ester linkage. But notice we can't just connect a bunch of monomers. Did you notice how we didn't do that? We didn't. There's not one monomer, and we're not linking monomer, 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 monomer. We just took a backbone and we added 3 fatty acids to it. Now, are the fatty acids long? Yep. But this cannot grow forever. In fact, it's limited to just those 3 carbons. That's what makes this not a MAC molecule. Okay? We call this a triglyceride. It's a fat molecule. Okay. So now I want to talk about these guys. Saturated versus unsaturated fat. We're zooming in and looking at that fatty acid chain. This is pretty common in the context of nutrition, saturated and unsaturated fats. And so saturated itself means full. So when we look at a saturated fatty acid chain, we have all single bonds here between our carbons, and every single carbon has 2, except for the end, 3 hydrogens at the end. Everything's stable, saturated, that fatty acid chain is full of hydrogen. When we come to unsaturated, what we have here is a spot where we've added a double bond. We still have to maintain adherence to that octet rule, right? We have to have that rule of 8. But when we added that double bond, we lost the 2 hydrogens here at the end. Okay? You guys learned in chemistry that molecules all have 3-dimensional shape. And so when we have this double bond added, we get a kink. Okay? It has a unique shape. So this is where I'd like to talk just very briefly about a phenomenon in the teacher world that happens from time to time. I give a quiz. We don't really do that in this class, but in other classes. I give a quiz. I hand the papers out. The kids take the quiz. There's always one that comes back like this. And I go, what happened here? I got mad. And I go, oh, gracious. Maybe there was drool on it. I don't know. So the student gives me back their quiz. I put it in the stack, and I end up with this. It doesn't sit nice and pretty anymore, does it? I can't get my paperclip on it. It doesn't go into my briefcase nicely. It's like bent. That one kink in the middle has made my whole pile of papers less dense. Would you agree that that's true? It's taking up more space than it otherwise would?
No.
That same thing is happening here. I have one kink, and it makes this whole thing less dense. They can't pack together as nicely as they otherwise would, because if this paper were still flat, my papers would fit together perfectly. This is saturated, but as soon as we add that other paper, where we have now space in there, that's unsaturated, less dense. We have that kink. It messes up the middle. Why does this matter? Because it affects how closely packed together those fatty acid chains are. So here, do we have any there that are unsaturated? No, saturated, saturated, saturated. Do they squish together pretty nicely? Kind of look like it, right? But what happens if there's a kink? So this is what we were just looking at, saturated. We get a kink, unsaturated. They can't pack together as well. So now I want you to think about this functionally. If you go to Albertsons and you pick up a thing of Crisco, solid, liquid, or gas, don't pick gas. Solid, it comes like white lard. If you pick up canola oil, solid or liquid? Liquid. The solid one is saturated. The liquid one is unsaturated. So next time you're sitting at home eating spoonfuls of Crisco, consider, that is true. Next time you're sitting at home eating spoonfuls of butter, consider you're putting solid fats in your body. They're saturated, which then means that once they get into your bloodstream, they are still solid, and that is what is causing heart disease. This is bad. Solid stuff is bad. The stuff that stays liquid, you pass, right? You digest and you get rid of. And so, because they're liquid in your blood, yeah.
So like, the saturated fats can't be like dehydrated, like they can't be broken up as easily, or they
No,
can't
they
be
so
broken
they
up at all?
absolutely can. So you can digest this. I don't want you to walk away thinking that when I eat butter, I have solids flowing through my veins. Okay, because that's not true. You can digest this. It's a lifetime of eating this instead of this that over time builds what are plaques in
Okay.
your arteries.
Yeah.
In fact, yep.
So like for metabolism, like so if people have fast metabolism, does that mean that they like digest things more? Like do they have those special things?
Is that for real?
No. Yes. So interestingly enough, what we believe to be metabolism is actually got to do with the floral and faunal palette of bacteria in your gut. The reason we say this runs in your family, right? People are like, oh, your whole family is thin. This is kind of gross, but we're going to go ahead and let it go. Like, oh, good. You get the bacteria that is in your gut that allows you to digest food from your mother during childbirth, right? So you have the same bacteria as your mom. It turns out that bacterial complex, that is what dictates your weight, okay? Right, so it's not about a fast metabolism. What we believe now, and this is a very high area of research, what we believe now is that it's actually the bacteria in your gut that actually give you these things. Yes?
So then what happens when you melt butter? Are you creating a cake?
Nothing, you're just changing the melting. All you're doing is like when you turn ice into steam, right? You're just adding kinetic energy. Yeah.
And sorry to interrupt, but like if I need to go to the bathroom,
I'll just go. Okay. You knock yourself out. And as it goes, you need to use the restroom. You help yourself. We're just going to keep right on plowing away. So there we go with fats. Let's talk phospholipids. You've heard of a phospholipid bilayer? Oh yes, that's in a cell membrane, right? Okay. So we're going to make a jump from nutrition to cell membranes. But I want you to see something. So I told you that with these lipids, we start with that glycerol here, right? And then we have 3 places to attach things. Well, on a phospholipid, on 2 of those locations, we attached a fatty acid. On the 3rd location, we added the phosphate. So what does that mean about what we have down here? This is a bunch of hydrocarbons, so this is going to be hydrophobic. What's happening up here? It's negative polar hydrophilic. Right? Yes?
Sorry, I just have another question. So since the like the fatty acids on the side are hydrophobic, that's why like butter it's like more unhealthy because it's harder for water to break it down. because it's like afraid of water, and water's the thing that helps you like digest.
I'll take your reason. I don't know that to be true, but your reason is sound. Okay. So, if you take a bunch of this molecule, phospholipids, let's say you take 1,000 of them, and you dump them in a bucket of water, what are all of your hydrophobic tails going to do? They're in water. Ah, water! We hate water! Wicked Witch of the West! I'm melting! So if I put a whole bunch of these in the water, these guys, this end likes water, this side hates water. What do you think they do?
They're gonna connect.
They're going to find each other and be like, oh, a friend. Oh, thank goodness, a friend. And that's what happens.
Wait, I thought you had dehydrated it to connect them.
No. So in a phospholipid bilayer that is a cell membrane, we are not going to physically connect your phospholipids. The only thing that holds together a cell membrane are hydrophobic interactions. The true loathing and hatred of our hydrophobic tails for water is going to cause them all to stick together with their friends so that both the inside and the outside of our cell, water here, and oh my gosh, I can't handle all the water in the middle. There are no bonds. Okay? It's just, I know now we understand what a cell membrane is. Steroids are also lipids. Steroids are also lipids. So, you gotta do those notes questions, man. If you've not been doing your notes questions, today's the day where you make that real. Steroids are also lipids. Steroids are characterized differently. So we're not looking at a glycerol, we're looking at a series of ring-shaped hydrocarbons. We can attach chemical groups to them, but anytime you see a series of rings like that, that's going to be a steroid. Examples are things like cholesterol and estrogen and testosterone. Cholesterol we hear about all the time because people have cholesterol that's too high, but the reality is you have to have some cholesterol because cholesterol is a precursor or used for things like estrogen and testosterone, which you have to have in order to survive. So that's not all. So here's what we're going to do.